All right. Thanks for being here, and thanks also for the intro, John. So this is the overview of the talk. I'm going to talk about, as John was saying, modeling of brain stimulation. So I'm going to start on how we model brain stimulation, and then move to why connectivity and recurrence is important, and then move to how stimulating the brain can reveal some of the physiology going on in the brain. And then I'm going to show you a couple of clinical applications. So first of all, let's start with the fact that there is a growing interest in both of these topics, both in whole brain model and brain stimulation. And this is a shameless advertising. We are going to host a symposium on Wednesday about this topic, and then let's start. So basically, what we want to do, we have a system which is like a node, and there are some different example of system, and we also have the brain, right? So we have some dynamics going on in this system. What we can do, we can perturb this system and basically study the changing in the dynamics and kind of the propagation of these dynamics. So in this actually, just a great study about from Andreas Spielberg and colleague. They use this model, which you know already, so the Janssen and Reed, and they basically inject an external input in the inhibitory interneuron, and they show how basically changing this input is going to change the dynamic, and specifically it's going to move to kind of a alpha oscillatory behavior to epileptic seizure behavior, and then if you increase further the external input is going to be just noise-driven activity. So, but this is still a single node level. And one of our interests was what is going to be if we inject the stimulus in one region and we measure that changes at the level of whole brain. So as a neuroscientist, from one side we have the anatomical connectivity, on the other side we have the functional dynamics, and the kind of relationship between these two entities matter of interest for several clinical and non-clinical purposes, and brain stimulation is just a way for unveiling these dynamics. So we have the connectivity, we have our brain, the connection, we perturb it, the brain non-invasively, I'm going to show you that in a second, and then we just study the functional dynamics that originate from there. So this is just a kind of some example of, of the brain stimulation technique available. Some are invasive, some are non-invasive. I'm going to show you example of those in the model framework during these talks. Again, we have the idea is that we have our model, which are our local dynamics, the one that we were also seeing during the hands-on session this morning. We stack these local dynamics in a parcellation, which we can decide, and then the connectivity between the parcel is modeled via the diffusion weighted image. And then we inject an input in some region, and then we study the dynamics that originate from there, and we can see some potential in regions that are the regions that are directly stimulated, which can be early latency or late latency, and then we can see also some propagation and some evoked potential, which are due to the stimulus that are propagating through time and space. So now we are gonna show you how connectivity is important in shaping this recurrence activity. One great example of how brain stimulation can kind of modulate the functional connectivities was due by this study when they basically model the TDCS, and specifically they use again the Janssen and Reed and the structural connectome to model the connectivity between the parcel. Then they use a TDCS field model to basically establish which region inject the input to, and then they inject the input to simulate the DCS effect in a canonical way. So this is a cathode-nodal montage. And then they simply analyze both the scalp EEG, so they took the source parcellation time series and they move it back to the EEG space, and exactly the same way that you did this morning during the hands-on session. And then they did the same, so they studied both the FFT and the functional connectivity for the scalp EEG and the cerebral area, so the sources. And what they found was like an increase in the functional connectivity in the regions that were supposed to, like, were affected by the TTACS. So this is the classic, as I would say, cathode-nodal montage, and we saw like a changes in connectivity. So, but this is still like at the level of kind of a global effect, because I don't know how much familiar we are with brain stimulation, but TDCS is not very focal, but induce like current pretty much in a, huge amount of brain. So one compelling way for actually injecting the stimulus in a kind of a high spatial resolution and in a non-invasive way is to combine EEG recording with TMS stimulation. And as you can see, we have like our ongoing EEG. Here is a TMS pulse, which is very focal, it stimulates a specific area, and then it basically generates some potential. This is what we call in jargon TP, so TMS-evoked potential. And there are like a lot of, pioneer study, very, very expiring by, for example, Maximini and colleague that have shown how this TMS EEG measure can be used in clinical practice, for example, for disentangle different level of consciousness, and also like other study by the same group when they show that, for example, during awake, we have a time series. So here is the EEG, here is the perturbation, TMS. As you can see, this kind of evoked potential are rich and are kind of, the stimulus is propagating through time and space. On the other hand, when we don't have consciousness, either because we are in coma or because we are sleeping, there is no propagation at all. So the system is showing these sleep-like waves. So it's responding to the perturbation, but then the stimulus quickly decay. So the cortex is not responding. So this is just a way for show you how we can use brain stimulation in a clinical context to disentangle different level of consciousness. So there are some studies that shown how basically we can model TMS in silico. For example, this study was very quickly simply like fitting the time frequency plot of 3 area when they inject the stimulus, and there was another study when they were measuring the PCI, so which is give you like a measure of complexity. And they were basically showing a good fit between the simulation and the empirical PCI. But these two studies were great, but they were not really showing like the spatiotemporal dynamic of TMS evoked potential. So this is our question, the question that we have. So we have the brain, we perturb it, there are some functional dynamics that originate as a function of this perturbation, and those are what is called recurrence, and recurrence is everywhere. So the kind of question that we have, it was like, what is the origin of this TMS evoked potential? We see some potential in some point in some space. Where does it come from? And there are like some studies that have shown that this recurrence activity can be modulated by, for example, a given task. And they show for these results for different spatial resolution, for example, non-invasively and invasively in humans. As you can see, there are comparable response, and they also show the same responses in rat using, again, non-invasive or invasive method. The kind of question, again, that we have is that where this recurrence came from, open the possibility to possible scenario. So on one side is that we observe some potential in a given space and a given time point, and this is simply due to like a echo of the perturbation. So it's the stimulus that is injected at a given space at a given time point and is simply like reverberated locally. The other scenario is a network reverberation. So the stimulus, in contrast, is traveling through space and time and bouncing back and eventually coming back to the stimulated area. So for doing that, for studying these two possible scenarios, this entanglement between two possible scenarios, we use like a computational model. Again, I'm not spending too much time on this model you saw already. Is the Janssen rate when the mu, in this case, which is PT, is the external input, we vary that and we inject the input in a specific area for mimicking the TMS. So that's the only difference compared to what you see like this morning in the resting state kind of scenario. So we have this model. What we did is that we stuck this model in a 200 parcel and 7 network coming from here. And then we model the connectivity between parcel with DWI, and we simulate the activity at the level of the sources. Then we move back to the space of EEG. As I was saying, I will show you like this morning during the hands-on session. And we got the first simulated TMS EEG time series. As you can see, at the very beginning, there was not a good fit between the simulation and the empirical one, which comes from like a open data set when the stimulus was delivered to the primary motor cortex. So we use, we cast the differential equation of the Janssen rate in a PyTorch environment. So we fit the parameter of the model using machine learning stuff for computing the loss and the gradients. And then after a few epoch or / iteration, we were able to come with a good fit between the simulated time series and the ground truth. So this is the approach. I'm going to show you a slide just to show you that the goodness of the fit. So this is our, so fit is good. It's like, I don't know, LT advertising. I don't know, but this is the first, the top row is empirical TP. Bottom row is fitted TP for 3 subjects. You see resemble kind of, no, the propagation pattern is highly similar. And this is the similarity for all the subjects. So now we can simulate reliably empirical TMS EEG data. So what we can do with that? We can answer this question. So we, as I was saying, we measure different potential in different regions, and we measure this potential via the EEG, so invasively. So what we can do with the model, we can create some lesion to the structural connectivity, so we can destroy the sum of the connectivity between parcels in a way that I'm going to show you in a minute. And this opens basically the option to the 2 scenarios that I was describing before. So from one side, there is like the local echo. So what happens, we can destroy the connectivity and nothing change, meaning that these responses that we see here are basically a local echo because the network is not important for shaping those responses. The other scenario is that on the other side, we create the lesion and we observe a suppression of the responses compared to the ground truth, meaning that this response right here is a network reverberation, because if we destroy the network, we are losing those responses. How do we create this lesion? So we simulate the activity, we identify the nodes that were more important for shaping the responses, and we simply cut their connection. And we cut their connection in 4 different conditions. So one condition was the control, so no damage at all. The other 3 experimental conditions, we destroy the connectivity 20, 50, 100 milliseconds after the TMS pulse. So those are the 4 conditions. Those are the propagation pattern. I know this is massive. I'm going to just highlight the key part of this slide. So if we stop at 50 milliseconds after the TMS pulse, so as expected, there are no difference between the no damage condition and 100 millisecond condition, because of course, in this case, the lesion has not come yet. But interesting, there is also no difference with the 20 millisecond condition. So in this case, the lesions came 30 milliseconds before this time frame, but nothing changed, meaning that this response that we observe here are completely locally driven, because if we lesion the connectome, nothing changed. So moving along in time, so on the other hand, again, here no difference between this condition and this condition, kind of expected, but differently than before, we see like a complete suppression of the responses for the condition when the lesion has already came in time, so for 20 and 50 milliseconds, you see we observe no responses anymore, meaning that this response that we observe here in the ground truth are due to like a network reverberation, because if we destroy the connection, those responses are completely suppressed. And moving along in time, again, 100 milliseconds, we have a mixture of the two effects. So we lost some propagation pattern to the ipsilateral and contralateral hemisphere. So this means that this is basically like network driven, but we still have some local echo, some reverberation, meaning that this response is purely local. So this is, okay. This is just a recap of what happens. So basically both time and space can shape the responses that we observe via TMS. And if we lesion the connectome, we observe what is similar to what I showed you before, so a sleep-like wave when we have no responses, especially for the late component. And I'm going to show you this. So yeah, this is similar to what I showed you before. For example, people found like in the context of anesthesia and coma, when we observe this sleep-like wave and the cortex is not responding anymore and the subject is not responsive to external stimuli. If we lesion the connectome, we observe the same kind of similar responses. Okay. So this is just another, this was like an example of the recurrence. So recurrence is due to like a perturbation and is basically like the connectivity is important for shaping these responses depending on what time we are observing that. There is also like this great study by Simone Russo when they show basically how stimulating the brain, basically this recurrence is due by the thalamus activity and the interaction between the thalamus and the cortex. So in this study, what they did, they basically like inject an external electric stimulation and then measure the activity. And then they did this also into a condition when they inhibit the thalamus via optogenetic in 2 different time spots. And then basically what they show is that this inhibition of the thalamus create basically a delay in the responses compared to when the electric stimulation is delivered without the thalamus inhibition. So the thalamus is important. And they use like a computational model for studying what level of interaction between the thalamus and the cortex are important in shaping those responses of the recurrence. So this is the results from the stimulation for this model when they inject the input in the pyramidal neuron. And as you can see, there are like interneuron, thalamic cortical neuron, and reticular neuron. And then for measuring what level of this model is important for shaping those responses, they create some virtual lesion, and specifically they create a lesion in the reticular neuron and nothing change compared to the ground truth. On the other hand, if they did the same for the interneuron, and they observe a completely changing of the response, meaning that the thalamico-cortical responses are mainly due like to this layer. This is not working. Okay, maybe it's this other one. Okay. So now I'm moving, so I show you like connectivity and recurrence. I'm moving how we can use brain stimulation for unveiling like brain organization. As you know, probably like the spontaneous resting state activity is not completely by chance, but is well organized in what we call like a resting state network. And in one study by, again, Andreas Spiegler and colleague, what they did, they used like a model for injecting the stimulus and study the resting state dynamic that originate from that. And what they show is that basically the injecting the input to one regions is basically able to evoke the resting state network connected to with that specific region. For example, here there are like the results of the simulation of one regions, and they were able to evoke the corresponding resting state network measured in the empirical data in the ACP. Another example of like brain states and how we can kind of use them for studying transition between one brain state in another was also already due by Joanna Scabral talk before with their approach about LIDA, and with this approach they were they want to basically try to kind of control the brain and promoting like some transition between a sleep state and a wake state, and basically what they did, they showed that basically like injecting the input and perturbing the brain in some specific area can kind of promote like a transition. So here in this case, they inject the input in some area and they were able to basically move the brain from like a state of sleep and to like a state of awake. And then this is the area that were more important like for driving this transition. So there are basically this posthaneous resting state. There are some state that we can measure using different approaches, and it's interesting that using the model we can kind of promoting a certain state transition compared to another. So in one of our study, we use like this framework and the network control theory framework for moving the brain from one state to another. And for example, in this case, we simulate brain activity, and we simulate like the TMS perturbation, and we stimulate the brain. We want to evoke like a specific resting state network. And for doing that, we use 3 different approaches. One approach was like the traditional stimulation site the people in general use in clinics. And on the other hand, we use like the path-lang. So this is the simply like a distance, Euclidean distance between the region. And then on the other hand, we use like a network control theory framework. And what we were able to show is that basically the control theory was network control theory was the optimal one for like evoking, in this case, the default mode network. So you see this, our 3 approaches. So like you see like with the network control theory, we were able to evoke the default mode network in its full form, so also like reaching a distant node. But this activity, it's organized and is not due by chance, but it's organized in a resting state network, and those resting state network are also like hierarchical, in a sense that had been shown several times in literature that we have some low order network, which are visual and somatomotor, which are characterized by functional segregation, and we have like a high order network, which are needs like the bottom of this pyramid for functional properly, and are kind of organized by functional integration on the other end. So the kind of question that we have is that what are the differences in the propagation pattern between this functional network, if any? So we use this data set, which is kind of amazing with our collaboration with the center of the University of Milan. There are 323 sessions, 36 patients, implanted patients, and we have both like intracranial electrode, and we record both intracranially and extracranially, and the stimulus was delivered like intracranially, and SPES stands for single pass electric stimulus. So there is a stimulus which was delivered intracranially, and we record both intracranially and extracranially. So what we did is that we identify what was the network that was perturbed, so what in the 323 session, and we divide the 323 sessions depending on what network was perturbed. So in this case, we were able to see differences between propagation pattern depending on what network was perturbed. So first of all, this is the grand mean average of all session. It's just to show you that it's kind of similar to what other, great group on working on intracranial EEG, like Joseph Parvisi, have shown like in literature, so with these three clusters. And those are the responses from the empirical data. So the key message here is that for when we perturb like high order network, the late responses specifically are higher compared to the low order network. So, and this is kind of similar to what people have shown, by great study, have shown like in literature how this, there is this hierarchical framework in fMRI community. This is like with EEG, and we show that both for intracranial EEG and extracranial EEG, so for two different levels of spatial resolution. So the next question is that, so basically this is just a summary. It's there are basically differences in how the stimulus is propagating depending on what network was perturbed. What the next question is, does this difference arise on different processes? So integration versus segregation. So we use basically our computational model for doing the same approach that I was saying before. So we create a virtual legion. In this case, we create the virtual legion for isolating the stimulated network. So the stimulated network can only speak with itself, but not with the rest of the brain. And the hypothesis was that if we do that for the lower level of this pyramid, so for the low order network, nothing change, or kind of unchanged compared to the ground truth. Because these responses, this network only rely on its own activity. The opposite scenario is that if we do that for the higher order network, so for the high order of this pyramid, we saw like a suppression of the response because these networks are characterized by functional integration. So this is, those are the results. And that's basically what we found. We found that for the interstructural connectome, this is the dashed line, compared to the disconnected connectome, we saw no changes for the lower level of the pyramid. On the other hand, we saw like a suppression of the responses for the high order network compared to when we run the simulation with the interstructural connectome. And those are, they are under the curve. So this is how much they change compared to like when we run the stimulus with the interstructural connectome. You see a suppression of the response for the high order network compared to the low order network, specifically for the late responses. Last part, I think I have 5 minutes, is just for show you a couple of brain stimulation model in the context of clinical purposes. So in this amazing study by Jill Meyer and colleague from Petra Ritter's group, they use this model, which is kind of a hybrid. So they also have like a subcortical population, and they have like so it's a spiking model for the basal ganglia, and there is like a neuromass for the cortex, and they have a couple of interfaces for connecting those two. They use it for modeling like DBS stimulation in the context of Parkinson's disease in this case. And the main results was that when they switch on in silico the stimulator, so the DBS, they were able to basically kind of let the response of the thalamus coming back to normality. So they show like a disinhibition of the thalamus, and it's interesting that this is what also like other people have found like with the Honeycomb model. So this disinhibition of the thalamus. And interesting enough, they were also able to, when they simulate, when they switch on the stimulation in the basal ganglia, they were able to evoke the resting state network that in general are like connected with the motor region. So as you can see, there is the primary motor strip, there is a little bit of frontal lobe, which is was kind of interesting. So last part, it's something that actually we are kind of doing at CAMH with brain stimulation and model of brain stimulation and in the context of depression. So the question that we have is that, as you might know, like TMS is a FDA-approved treatment for depression, rTMS, and there are like a lot of great studies that have shown how basically we can use functional MRI, for example, for predicting the clinical outcome or TMS-EG measure. The question is that can we use the model for predicting the responses in a kind of making the model clinically basically helpful in some case. So we work with this with data sets. So we have like a TMS EG cohort of depression people. So we have these people with resistant depression. We did like a clinical assessment and then a single pulse over the DLPFC. Then there was like the standard ITBS treatment for 30 days. And then we have a post kind of evaluation which was similar to the pre. We use the model for modeling the pre and post of the DLPFC single pulses, and what we found is that the excitatory feedback loop of the model was predicting between responder and not responder, and specifically we saw like a kind of a shift compared to the pre. So this is the average at pre of the excitatory feedback loop. You see that for the responder at the post tend to be increase compared to like the not responder. And interesting enough, we also study like the state that came out from the model. So this is actually the brain state of the neuromass that predicted the clinical outcome, and was interesting to us that was similar to like this SGC and the LPFC negative correlation that people have found to be predictive in the fMRI community. So what we did, we took these brain states and we plot like the functional dynamics for the responder and not responder, and this is for pre and post. At 0 there is the TMS pulse, and this is how the basically the state is evolving through time and space for all these fixed point. I'm gonna screenshot this movie here. So what we measured was that, what we noticed that there was like more difference between pre and post between these dynamics for responder and not responder. If we measure that via like pointwise Euclidean distance, we found that the difference was mainly located like here in the kind of a late TMS-EG responses, which is also like coherent with group of Davide, Diaconesco's group, what they shown like in the context of TMS-EG data. And with this being said, I'm just concluding and say that hopefully this brain stimulation model can be kind of useful like in the future. The idea was like to move the brain and to tailor the external input in a way that we can promote like a healthy target brain state. This is just a recap. I show you like how to model brain stimulation, how connectivity and recurrence can happen, and how brain stimulation and model of brain stimulation can be used to unveil some physiological brain organization and some like clinical application. With this being said, everything that I show you is replicable. So there are like in our GitHub repo like for every study that we are running. And just want to say that we have a few posters in the next few days. We will be grateful if you would like to stop by and give us some good feedback. And with this being said, I just want to thank all the people that contribute to this study, and also like our group, which I am very fortunate to be collaborating since 3 years now. And thank you.